So to begin with, if you want to do a nice warm-up question, why don't you introduce yourself to us?
That's a nice warm-up. My name is Rhys Jones. I'm the Technical Support Director for Aber Instruments. I've been with Aber for about 10 years now, which sounds crazy when you say it like that. I started as a product application and lab specialist, and have then kind of stayed in a similar customer-facing role in that time. The tech support function oversees, as the name suggests, all technical support functions, but in Aber, as a relatively small team, also oversees field applications work, lab responsibilities, and services as well. So it's quite a diverse range of responsibilities within tech support.
Yeah, during the introduction part, I did mention how you've been working with Aber for many years in the technical support team and now leading the team as well, and that you've traveled quite a bit representing our technology. And you sort of end up seeing so many different applications, right? And inadvertently end up seeing our technology being used across different scales and different platforms. Do you want to maybe talk about that a little bit? Just the like what I think is a privilege almost.
I was gonna. The first thing that sprung to mind was that word exactly, because I never would have thought that this small, humble company like Aber, or at least that's what it was when I first joined. We're much bigger.
It's not humble anymore.
As soon as present company. It was a relatively small team. We're a much larger organization now.
Yeah.
Still maintaining sort of those humble roots, but it just seemed bizarre to me that this company operating in a small town in Midwest Wales was partnering with companies across the world, and just had these incredible setups and going into such important processes. It really was humbling to see that, and you wouldn't expect it from the kind of organization that we are, the kind of people that work here. Incredibly impressive individuals who do what they do, but then you just wouldn't expect the technology to end up in the places that it ends up, and in the hands of also amazing people who do incredible things with it as well.
It's so true. I mean, at last count, we probably have supported more than 150 countries or so where our technology is being used.
I think over that, yeah.
Which is, again, like you say, quite humbling and almost shocking. So yes. So when we travel, we get to work with amazing companies, individuals, groups doing incredible things with our technology, and just to produce drugs and other products that are hugely beneficial to mankind as a whole. So let's sort of bring back the topic on scalability then. Let's just start with the basic question: is Aber technology scalable?
In a word, yes. we could end it there. No, we recognise that at Aber that scalability is massively important to our customers, and as a result, it's incredibly important to us, and it's really at the core of everything that we do here at Aber. It's also part of any new products that we develop, as well as every product that we have developed historically. We always strive to ensure that all of our products are scalable and comparable with one another. We have a really diverse product range, and I think that reflects the diversity of the bioreactors and environments in which our sensors end up with in the market. So there's a large range of form factors, electrode configurations, materials used in our sensors, but invariably with all of these, the the measurements themselves are identical across the board and scalable across the
You
board.
raise a very good point, actually, because uh, when it comes to scalability, of course, it is, I feel, our responsibility to offer solutions that work across different bioreactor volumes and form factors. But also, whenever we develop something new, I know this for a fact because, fortunately, for me, I've been involved in many of these development projects, and the URS that we come up with always has a point about the measurements being comparable to everything that has come before that product, which I think, like you said, we take quite seriously because that is one of the first questions that our customers ask us.
Yeah, no, I think the thing that's impressive is that it almost doesn't need to be said.
Yes.
It's at every level in Aber.
Mm.
It could be unspoken. Of
Yes.
course, it's in the URS, but everyone knows and understands that the measurements have to be scalable. They
Yes.
have to be comparable to everything that's gone before, because that's what matters to our customers.
Yeah.
So yes, it always ends up in the URS, but if it didn't, then everyone understands that needs to be the case, and that is the case.
Yeah. Excellent. So the next question is simple. Why do I believe you? Which is, the proof is in the pudding. We work in the scientific industry. We work on evidence. So the next question, if I were to put it the other way, is what evidence do we have that our technology is scalable?
Absolutely. And I think the answer that I would give to anyone who we spoke to, anyone outside of Aber, is that really you shouldn't believe us.
Yes.
I think inherently the industry that we work in is inherently skeptical and wants to see the evidence for themselves, and rightly so. So I always encourage people to try the Aber systems. I think that's the best thing, and the proof is in the pudding. Is to try it for themselves and to validate it themselves.
Yes.
And to build that trust and confidence in the Aber products.
Yes.
But having said that, we do a huge amount of internal validation as well as external validation with partners, but also independent third parties have conducted a lot of studies into this over the years. So some notable examples of these would be Sartorius, who conducted a study on looking at the scalability of the biopack biomass. The biopack biomass, for those who aren't aware, is a co-developed, dual-branded Aber Sartorius product. So it takes the underlying core Aber technology, and that's directly integrated into Sartorius's control system. That's integrated into the rocking motion platform as well as the STRs. And Sartorius conducted a study where they went from the 50-litre rocking motion platform up to the 2,000-litre STRs. And in that, I'm sure we'll be able to show a graph or a graphic on screen. They demonstrated near perfect correlation between these. I think in the data, the capacitance goes up to about 100 picofarads per centimetre, within the Aber range. I think that probably roughly equates to maybe 100 million cells per milliliter, something in that vicinity. And there are these characteristic dips that are seen throughout the process. These are just the dilution effect from it being a fed batch process. But in every single one of these platforms and scales, the capacitance is highly comparable throughout. And to the point that we discussed earlier, that not only gives users the confidence that the technology is scalable, but also it can be used as a key performance indicator to show the user that your scale-up strategy has been successful.
And if I remember, that is a good data set to talk about. If I remember correctly, that study was also done across different sites at Sartorius. It wasn't just performed at one site. So again, in terms of, and this often happens even with our customers, right? Like depending on the facility, they may have the amenities to perform their experiments at a certain scale, and then to perform the runs at another scale, they have to go to another site or facility. And I think that's what Sartorius did as well for this study. And uh, if it is the data set that I think you're talking about, then the capacitance trends did sort of match and sort of sat on top of each other. But also because it's a real time measurement, it was also able to identify critical events in the process. And in one particular case, it observed a deviation in one of the scales that was completely missed by their offline counts.
That's right. Yes. So I believe in the data set you're referring to, there's a sort of a peak. There's one
Yes.
of the trends is a slight outlier, and I think that was due to an individual operator error. But again, highlights the value that capacitance brings with this real time measurement in that it's able to catch, identify, and they were able to rectify this issue before it would have appeared in any offline counts. So kind of the real value of capacitance is in the real time live in
Yes.
situ measurements that it's giving you of your viable cell density.
We are, like you said earlier, we are quite privileged now because our technology was invented back in 1988. So it is now a relatively mature, well accepted solution in the industry for cell density measurement. And that has only happened because of these independent studies, and very often that have been published in peer reviewed journals. So yeah, so we sort of have that sort of wealth of publications in our pockets. But like you say, our approach always is to provide as much information as we can to our customers. But then our final, essentially, our final say is try it for yourself and see. So the Sartorius case study was a fantastic example. Any others that come to mind?
Yeah, absolutely. So as you said, Aber is a mature technology, and the benefit of that is that it's been widely used and adopted, and as a result, there's a wealth of publications available, which we'd be able to refer any listener to. But one particular notable example that springs to mind is that of Biogen. So Biogen conducted a study where they looked at capacitance against their viable cell density in 5 litre, 200, 315, and then their 15,000 litre manufacturing scale. And in that, they observed a near perfect R squared value, so an R squared of 0.99. So really near perfect correlation between capacitance and viable cell density. Biogen are a particularly interesting case study here because they've, since I think 2016, have adopted Aber as their gold standard. They no longer convert capacitance to VCD. Of course, many users continue to do this, and that's, capacitance is highly comparable to VCD, and many of our users choose to do that. But the option is open to them, as Biogen have demonstrated, that you can also trust and rely upon real-time capacitance solely without the need for that conversion. And so Biogen have since then started utilizing capacitance for control of the feed of their complex nutrients. They utilize capacitance for troubleshooting their process, and a range of other applications on top of that. So it really, they really have been power users of the Aber systems.
Yeah, Biogen, I mean, there's so much to, when you mention the name Biogen, there's so much to talk about. Even their work on sort of demonstrating that capacitance is more sensitive to apoptosis as compared to offline measurements, typically type in blue measurements. And I believe even the scalability study that you were talking about earlier, they compared capacitance to their offline data. And just a quick sort of caveat that graph talks about only the exponential phase of growth, which which they were most interested in at the time. But yeah, so like I said, yeah, Biogen, one of my most favorite studies of theirs, which I spoke about in a webinar recently too, is how they started with the premise of, well, why do we um, feed our cells once every 24 hours? And of course, the answer to that question is, historically speaking, feeds have been sort of based on offline counts, which are done once every 24 hours. So here we have a technology that's giving us results once every 4 seconds. So Biogen said, well, what would happen if we were to feed our cells more often? Because we have the ability to do that now. So they ended up sort of testing a scenario where they were feeding their cells once every 4 hours instead of 24 hours, and immediately they demonstrated how this new strategy, compared to their manual 24-hour feed strategy, resulted in better cell growth, did not result in glutamate depletion, and improved productivity. The top of my head, was it 21% or some? Quite an impressive number, really. Anyway, so I know we've sort of diverted a little bit.
No, but I really love that example because it's another example of customers or Aber users utilizing capacitance for things that I never would have
Yes.
predicted that they'd be used for. So that there are so many diverse ways in which you can utilize capacitance, and our users keep finding new and interesting ways of doing that. You know, challenging conventions or current
Yes.
conventions, and finding, you know, optimizations that can be made.
Yes, I mean, and that is
amazing.
that's why it's so sort of it's so interesting to be working with customers like these, because, like you say, things have been done a certain way for a long period of time. But I think the joy of science is to sort of understand what you currently have and what you can change for the better, right? And I think a lot of our customers, Biogen included, have demonstrated that. Okay, so we've covered a few important topics already. So we've talked about the Sartorius case study, which focused mainly on our single-use biopat biomass solution and how that is scalable across the rocking motion bags and the STRs. Then we spoke about the Biogen case study, where they used our reusable capacitance solutions across different scales through to manufacturing. But how about, are there any studies that compare our reusable with our single-use solutions?
Yeah, absolutely. So a great example of this is through our partnership with Thermo Fisher. So much like the relationship that we have with Sartorius via the biopat biomass, we have developed the Futura Neo alongside Thermo Fisher, which is integrated into their high performer bags. And as part of the validation of the Futura Neo, it was tested thoroughly alongside the Futura, the reusable Futura, in the high performer bags. So a number of studies were done at Thermo Fisher as well as at customer sites, so commercial development partners such as Biogen.
Yeah.
To compare the reusable and the single-use range. And I think it was in the 50-liter scale. The capacitance was compared between the single-use and reusable. And in that, the capacitance trends were highly comparable. I think there was 2% or less than 2% variance between the two trends. Across a number of different case studies, or across a number of different runs, which is really quite impressive.
Yeah, and if I remember correctly, Rhys, the study that Thermo performed, they had 3 Neo sensors in the same bag, along with a reusable annular probe installed via a clean pack connector or something similar.
Yes, either clean pack or a CPC aseptic quick. That's right. So, you know, we've got a number of data sets, and it wasn't isolated to, you know, it was multiple runs conducted at Thermo and with commercial development partners. And this consistency of trend was seen across across multiple sensors.
Mm-hmm.
So yes, absolutely. That's again kind of going back to the original question about scalability and the development
Mm-hmm.
process here at Aber. Any new product
Yeah.
always has to be scalable and comparable in its measurements
Yeah.
to the current range, and then again traceable back to national standards as well.
What was also encouraging was that when Biogen um, performed their runs with the Neo, they actually tested it in both fed batch and perfusion applications, and even with perfusion, they tested it at two different set points as well. I believe it was two different set points. Yeah, it was tested really very thoroughly, and I was quite pleased to see such good data coming out of that. Back then, the newly developed Neo sensor. Earlier, you also discussed, Rhys, how Biogen was using the raw capacitance profile to feed their complex nutrients automatically. Uh, I want to sort of flesh that point out a little more. How do you think our technology has been used uh, to control critical events during the scalability?
So kind of going back to a couple of the points that we've talked about earlier. Firstly, the maturity of the technology, but also the diverse and interesting ways in which users utilize
Mm-hmm.
capacitance technology.
Mm-hmm.
There's a really diverse number of ways in which our users do utilize capacitance.
Mm.
But the starting point generally that we would recommend to any user is to begin with just monitoring your process.
Yes.
Starting with the basics, just monitoring your process, and it can really be quite enlightening when you first go from looking at single data points 24 hours apart,
Mm-hmm.
and kind of giving you an idea of what's happening in your process, but not a complete picture.
That's right.
And then transferring that across to measurements every 4 seconds,
Mm-hmm.
building that real-time picture of what's happening throughout the course of your project, of your process. Once that's been done, then generally users might then start to build a batch fingerprint or a golden batch for their process. And then you can start to do interesting things with that. So you can start to build a sort of standard deviation around that batch fingerprint and utilize that for troubleshooting your process if it deviates from that golden batch. You can start to look at ways in which you can optimize your process, optimizing feed points, optimizing harvest, and then you can get into the real core value of the technology, which is utilizing capacitance for process control. And that's where we always encourage our customers to get to, and that's certainly where our users get to when they utilize capacitance in cGMP environments.
Yes.
Is utilizing it for process control, and you can do so in a number of different ways. We've talked about Biogen utilizing it for the control of feed of complex nutrients, but it could be to control harvest point. It could be to control the cell concentration itself through perfusion applications. And these are the real value additions
Yes.
that capacitance can provide. And there's a really nice, diverse range of applications outlined on the Aber Instruments website. So there's an applications section which kind of highlights the diversity of the applications which our customers have utilized in the past.
It is you mentioned perfusion. It's been very interesting for me to see the evolution of Aber capacitance's use in these applications, because initially it was, like you said, for monitoring, and the control was done with something else. Over the years, it's now become almost a standard application where you control your perfusion rates using capacitance. With that said, there's a school of thought where you still want to convert capacitance to cells per milliliter, and then you use that information to control your perfusion rates, still the cell specific perfusion rate. But then in the recent past, we have an example from Lonza, for instance. They published a paper a couple of years ago where for the very first time, I believe they've coined the term cap SPR. So they control the perfusion rates based on capacitance specific perfusion rate. And I feel it's this particular application has become so mature that several users have reported um, media cost savings when Aber capacitance is used for controlling perfusion rates. So we have the likes of Janssen and Lonza and even Sartorius reporting, media cost savings from 14% to 40 or 49% in some cases. And why I feel this is why this is rewarding for me to sort of observe is that perfusion is great for many, many reasons. All of us are aware of it. Continuous manufacturing, process intensification, and that's where the industry is headed. But one biggest hurdle of perfusion is media costs, right? Because you end up, feeding your feeding your cells continuously. So you end up using a lot of media, and media is not inexpensive. But if you can utilize the Aber technology then to overcome that hurdle, then that is, like I said, quite rewarding, a thing for me to observe.
Yes, and I suppose that comes back to the underlying principle of the Aber measurement, which is that it's a reflection of the membrane-bound biovolume
Yeah.
of the culture. And so you're feeding your cells based on the volume of the cells present rather than the cell number,
Yes.
which one could argue is probably a more accurate
True.
reflection of the nutrient demand of that culture or
Yes.
that process.
Yes. Now this goes back this goes back a few years, but one of our customers actually gave me this analogy or an example. They said that if a normal, like let's say an average man, would need a set number of calories a day, let's say 2,000, 2,500 calories a day, but if that man were to be the previous like world sumo champion Byamba, that's the example that he'd given me, a specific example. He would need a lot more calories to be doing his job correctly, which is essentially to push people out of the way.
It's an important job.
But that was a good example, I thought, to demonstrate how, depending on the volume of the cells, the nutrient requirements for those cells would be different too. And like you said, capacitance is effectively a biovolume measurement. So you end up feeding your cells more accurately in that sense. Yeah, that was good. Just to go back to first principles again, Rhys, just if you don't mind, when it comes to scalability, we talk about many of our customers ask, well, what would happen if I use the Aber probe in very small scales and then uh, through to very large scales? And obviously, we've discussed data in this chat that demonstrates that it is actually scalable. But talk to me about the electric field of measurement as well of the different probes that we offer and how that plays into scalability.
Sure. I touched very briefly on the different form factors, electrode configurations, different materials used in the Aber sensors, and that being a reflection of the diversity of bioreactors available on the market. But in particular, the electrode geometry,
Yes.
and that kind of dictates the field size. That is sort of pivotal in this scalability discussion, because for probes which are intended for small benchtop bioreactors, the electric field is designed to be much smaller, so probably the size of a marble in the cases of some of our smaller single-use probes, and then going up to the size of a tennis ball. But all of these, regardless of the solution that they're in and what they're actually measuring, the measurements themselves will be highly comparable regardless of that. So again, it's the field size is almost not inconsequential because it is important, but from a user perspective, it's inconsequential because the important thing is that the measurements themselves are the
Yes,
same.
yeah, and there's a couple of things there, right? Even in smaller scales, if it's certainly very possible that the tennis ball electric field could be hampered in sort of tighter spaces. However, uh, I think most of our users would be aware of this, but we sort of zero the probe in cell-free media before inoculating the cells, which means you're sort of taking away the background capacitance of cell-free media, almost like tearing a weighing scale. So you start with the same sort of capacitance each time. But the other thing as well is, and this is something that is not spoken of enough, that the unit of capacitance measurement is picofarads per centimeter. And that per centimeter piece is what makes the measurement universal. It takes into account the geometry of the electric field.
That's right. So during our in-house calibration process, that conversion from these raw or raw farads, raw picofarads,
Yeah.
to give it a dimensionality, that a probe constant is applied to convert it to picofarads per centimeter. And that measurement then is traceable back to national standards. So if you were to measure two probes with completely different electrogeometries, different field sizes, if you measure them in conductivity solution from a third party, which is traceable back to national standards, the measurements in both should be exactly the same.
Excellent. I think that covers it quite nicely. I think we've covered quite a few topics around scalability in this discussion. More importantly, we've spoken about some data sets that come from independent studies that demonstrate scalability, and also, as you correctly pointed out earlier, just sort of demonstrates how Aber technology can be used as a performance indicator to understand whether the process is in itself scalable, or the scaling strategy has been successful. And we also spoke a little bit about how to get the best benefit of Aber measurement, you should be looking to control something critical in the process in addition to getting a real-time fingerprint of the process, which in itself is quite valuable as well. Yeah, no, thank you for that chat, Rhys. Really enjoyed it.
Thank you very much.
Thank you.